All right, this is Unit 6, Part 2. We're going to continue looking at the heart. In Unit 6, Part 2, we'll look at cardiac physiology or function of the heart. So let's talk about the valves. There are 4 valves in the heart. What they do is they make certain that flow, their job is to make certain that flow is unidirectional, that there's no backflow at any time. So they prevent flow backwards against the normal direction of the flow of blood. There are 2 AV valves, atrioventricular valves, between the atria and the ventricles, one on the left side and one on the right side. And they are the tricuspid on the, I'm going to go with right, and bicuspid on the left. What happens, of course, is that they're open when blood is flowing from the atria through into the ventricles. And when the ventricles contract, they snap shut. There are these connective tissue strands called the chordae tendineae that anchor those AV valves. Chordae tendineae are attached on one end to the valve and on the other end to the papillary muscles. So when the papillary muscles contract along with the rest of the ventricles, they're pulling on the chordae tendineae and preventing the flaps from reverting upwards, getting pushed upwards by the pressure of the blood in the ventricle, and so they don't get so they don't allow the flaps to open up backwards and allow blood to flow from the ventricle back up into the atrium. And you can see that here. What you see here are the the papillary muscle. The papillary muscle as an extension of cardiac muscle tissue from the wall of the ventricle on both sides, attached to the chordae tendineae here, which are in turn attached to the flaps. And so when the ventricles contract around the blood that fills the chamber, then the papillary muscles also contract and the valve flaps get pushed up and closed, and they're held in that closed position and can't actually flip upwards like this. Let me change color. They're not going to be able to flip upwards. Here the flap, there's the flap. When it's closed, it won't be able to flip upwards and allow blood to flow back up into the atrium, because the chordae tendineae are holding them in position. And the closed position, of course, I'll draw it for you. The closed position would be like this. One here and the other one there, right? Oh, and on this side it would be here and here, like that. So these chordae tendineae here are pulling down, and the analogy that's always typically used to describe how they work is that the papillary muscles are like the arms of a parachutist who's holding on to the cords of the parachute. The cords of the parachute are represented by the chordae tendineae, and the valves are the parachute itself, the dome of the parachute. Okay, again on the, as I said earlier, on the right side it's the tricuspid, and on the left side it's the bicuspid, which also is often called the mitral valve. Papillary muscle, chordae tendineae, etc. There's quite a few things on this that you would have to know. In fact, almost all. Okay, the other set of valves are the semilunar valves, which prevent any blood getting back into the ventricles after the blood is ejected out into the aorta on the left side and into the pulmonary trunk on the right side. So the aortic semilunar valve is between the left ventricle and the aorta, and the pulmonary semilunar valve is between the right ventricle and the pulmonary trunk. So when the ventricles contract, the AV valves, the tricuspid and the bicuspid, close, and there's actually a period of time when all valves are shut and you have an increasing contraction of the ventricles so that the pressure builds and the semilunar valves snap open, and blood in the left ventricle shoots up the aorta and blood in the right ventricle shoots up the pulmonary trunk. Okay, this is really this is a drawing of an angle cut. It's a cut across through the heart. It's an oblique, which means at an odd angle, not 90 degrees to anything. It's an oblique cut across that shows all of the valves, that's all. This is quite a nice photograph taken inside the chamber of the heart. What it's showing is the right side of the heart, in the inside of the right ventricle, and what you see are the papillary muscles and the projecting up from the wall, holding on to the chordae tendineae, which are attached to the flap, the valve flap. I'll do it in a different color, the valve flap here, and it's a little difficult to see where it is. I guess in somewhere in, I don't know, here and here, holding onto the valve flap of the flaps of the tricuspid. One thing you should note in this photograph of the heart on the right side of the screen is that the wall of the left ventricle is much thicker all around the left ventricle. It's much thicker than the right ventricle, and the reason is because there has to be much more pressure generated to eject blood up into the aorta and then through the bicuspid. I'm sorry, not the bicuspid, through the aortic semilunar valve, so that it goes out throughout the body. And that contraction of that very thick muscle on the left ventricle wall, the left ventricular wall muscle, actually causes a pressure wave to go throughout your arterial side of your systemic circulation. And you can feel evidence of that pulse, of that pressure pulse, when you take your pulse at your wrist. That represents each beat, each contraction of the left ventricle. On the right side here, on the right side, it's thinner because it only has to send it up to get up to the lungs, and then it gets oxygenated and comes back to this side, the left atrium. So in terms of valve function and the cardiac cycle, you have blood. I'll do it in red. I think that should work. You have blood coming back into the right atrium through the superior vena cava, and also here through the inferior vena cava, and of course some blood coming, and it's not shown, the opening's not shown, but coming from the coronary sinus. And actually the valve is, the AV valve on this side, the tricuspid valve, is open, and as the blood flows into the right atrium, it also spills down into the right ventricle. So you get filling of the atria and blood spilling into on the right side there, and blood spilling also into the right ventricle. And then that's when the heart's relaxed. And then first the atria contract, and then the ventricles contract. The right and left atria contract at the same time, and then just a split second later, the ventricles on both sides contract. So it's always the superior part of the heart that's contracting first, followed almost immediately by and then it starts to relax, and then the ventricles contract. So as the ventricles contract, now the tricuspid valve here shuts, and the aortic semilunar valve is still closed, and you have an increasing pressure, and the pressure builds, and then the did I say aortic semilunar? I meant pulmonary semilunar. And then the blood surges out through the pulmonary semilunar valve and goes up to the lungs through the through the left pulmonary artery, and it's not shown, but it would be back here through the right pulmonary artery. Okay. Now, so here, this is pressure building, and I guess on the next slide it'll show the blood being ejected out. Yeah, so here you go. Now, when pressure builds high enough in the ventricles, the same thing, by the way, is happening on the left side. Pressure's building there. This valve here, the mitral or bicuspid valve, gets snapped shut. Pressure builds up in the left ventricle, and then the blood eventually, the pressure is high enough to open the aortic semilunar, and the blood rushes up the aorta, goes throughout the body. At the same time, on the right side, pressure builds, and then the pulmonary semilunar valves, which are here and here, they open up, and blood rushes up and out through the pulmonary trunk and up into the left pulmonary artery and the right pulmonary artery. Okay, so we have blood rushing out, and then it's followed by a relaxation of the heart, and that's shown here in this diagram. The heart relaxes. There's a bit of a back pressure wave. So blood is shot up into the pulmonary trunk and the aorta. There's always a bit, as it shoots up into that narrower space, you get a bit of a rebound off the wall heading back, and that helps actually shut the valves, so preventing any further flow. There's just a little bit shock wave coming back, and you can see that happening here. The wave of pressure comes back, and these semilunar valves, and they're called semilunar valves because they're a half of a sphere cupped upwards, they'll snap shut. They'll catch that wave of blood bouncing back off the walls and snap shut. Okay, let's look at the cardiac muscle tissue at a microscopic level. Cardiac muscle is one of the three subcategories of muscle tissue in the body. The other, of course, are the skeletal muscle and smooth muscle. Cardiac muscle, of course, is involuntary. You don't control it consciously, so it's unconsciously controlled. It is, as I told you before, striped or striated, relatively short, unicellular, branched. At the ends of the branches, you have intercalated discs that have many gap junctions that allow flow of the flow of ions directly between the different cardiac muscle cells. Cardiac muscle cells have a lot of mitochondria, and therefore, well, they're constantly, the heart is tissue is constantly at work, needs to generate a lot of ATP, therefore needs a lot of oxygen. The intercalated discs, as I said, help anchor the cardiac cells together at the ends of the branches and allow free passage of charged particles or ions because they contain many gap junctions. The heart muscle acts essentially as a single fused cell, a single unit, because of this free flow of ions between the cardiac muscle, individual cardiac muscle cells. At least the top part, both atria, will act as a single unit and all contract simultaneously. And then there's a delay, a short delay, and then both ventricles contract. The purpose of the atria contracting is to eject whatever blood is in them down into the ventricles. And the purpose of the ventricles contracting is, of course, to build up pressure and then shoot it out into the aorta on the left side and into the pulmonary trunk on the right side. A single unit of cells is a syncytium. And I described to you a syncytium of skeletal muscle cells occurs during a fusion of skeletal muscle, or syncytium of skeletal muscle cells occurs during development in the embryo, so that skeletal muscle cells actually fuse into a single very large, long, multinucleated skeletal muscle fiber. You don't have skeletal muscle cells, you have skeletal muscle fibers, and they're multinucleated. They're formed by a true fusion. The heart muscle, at least the atria, and separately the ventricles, acts as a functional syncytium. In other words, it functions as if it was a single fused syncytium, fused cells, altogether fused into one unit. Here you see a photomicrograph on the upper left here of some cardiac muscle cells. You can see the single nucleus, branched appearance, striated appearance. You can easily see the, let me do a different color, many intercalated discs at the junctions between branches. Of course, you have to remember that this tissue has 3 dimensions, not just 2. It's not like a flat piece of paper. It has depth, further depth to it. So you don't see the nuclei always, but either in front of the screen or behind the screen, there is probably a nucleus here and probably another one there. It's just that this plane of cut did not cut through like it did in this one in the middle. All right, and here you see a drawing of an intercalated disc in a larger magnification, a drawing of an intercalated disc here. And you see that there are gap junctions allowing for passage of ions through. Here's another view of, actually here in this diagram, you see the gap junctions here, embedded across here. So, contraction of cardiac muscle tissue is stimulated in 2 ways: by external innervation, nerves that innervate, bringing impulses to the heart, but it is also self-excitable. The heart is said to have automaticity. It is self-stimulatory. It contracts as a unit, at least first the atria on the top of the heart, and then just a small short, with small short delay, the ventricles below. The contraction of the heart has what's called a refractory period. It's a short period, well, relatively long relative to other kinds of tissues. Refractory, I'm not going to get into that. I'll describe that later. Okay. Okay, in terms of the autorhythmic ability of the heart to generate its own impulse that allows it to contract steadily, even in the absence of any external nerve supply or innervation. There is a small amount of tissue within the heart that sends out steady impulses, and those impulses spread down throughout the atria and down also into the ventricles through this conduction system. And the impulses are initiated at a particular area, which is located in the wall of the right atrium. Now, I have to explain to you how these impulses occur, and first I'll explain a few things about impulses occurring in other types of nerve or muscle tissue. The fact is that if you have separation of ions across cell membrane, then that is what's called a membrane potential. So if you have separation of ions where there's different concentrations of ions on either side, so there's a difference in charge because there's more negative ones on the inside than the outside, for example, then the inside will be, because of that separation, that the inside is said, the cell is said to have a membrane potential, and the potential is measured in millivolts. And all cells, in fact, are more negative on the inside than the outside. So if we look at the voltage, then you'll see, and you're looking at most cells, other than the autorhythmic cells that reside in a small place in the right atrium, most cells, if this is the graph, most cells have a resting membrane potential in the negative range, okay, and it might be minus 60 or minus 40 millivolts. You'll see different values, but it's a negative resting membrane potential. And if they get impulses, and this is only true for muscle tissue or nerve tissue, if a nerve impulse arrives onto this tissue and causes channels to open in the membrane that allow passage of ions, then what you get is a sudden change in direction towards the more positive. Okay, and it comes up and it spikes up, and that rapid change is called an action potential, and then it quickly reverts and comes down. This is going upwards is called a depolarization, and coming back is a repolarization, and there's a bit of an overshoot usually, and then it comes back up to the resting membrane potential. But this is all dependent, this is in millivolts, okay? And this across here, this axis across here is time. But this is all dependent on arrival of an electrical type of nerve impulse, okay? Unlike this situation, remember that this rise is called a D. I'm not going to spell it out for you twice, but this is called a depolarization. And this is called a repolarization as it comes back down. You know, it's polarized and then becoming depolarized on this side, and going back to being repolarized on this side. So depolarization and repolarization is occurring. Normally the cell is sitting there at a resting membrane potential, negative relative to the outside. Then it receives an impulse that causes it to go more positive. Again, this is measured on the inside of the cell, and it actually shoots up into the positive range. That's a depolarizing effect, and then it repolarizes and even overshoots down below the normal resting membrane potential and returns to the resting membrane potential. Okay. Now the heart, that is different. This, by the way, this spike here is called an action potential. This depolarizing, repolarizing wave is called an action potential. Unlike most other tissues that have what I've just drawn, that kind of response to external stimuli from external nerve impulses, the heart has specialized autorhythmic cells that can initiate their own action potentials. Their resting membrane potential here, in this case with most other muscle or nerve tissue, their resting membrane potential is not stable. It is unstable. These specialized autorhythmic cells have the following characteristic, and I'll show you. So, unlike what I drew on the previous slide, which was for other types of muscle tissue, except for that small amount of autorhythmic pacemaker muscle tissue found in the heart, all other types of muscle tissue have that kind of characteristic that I just drew on the previous slide, and that also goes for nerve tissue. So nerve tissue and all other muscle tissue, that means skeletal muscle, smooth muscle, and the vast majority of the cardiac muscle tissue, with the exception of pacemaker, autorhythmic tissue in the wall of the right atrium, they all have that kind of characteristic resting membrane potential that needs an external stimulus in order to generate that spike of an action potential. Now, the difference in the autorhythmic tissue is that what you have is an unstable resting membrane potential. It does not stay at a flat negative value. It is constantly depolarizing. Gradually, it depolarizes up to what is called a threshold value here. That's the threshold. And once it hits that threshold, then you have a rapid generation of an action potential. And the action potential is this spike here, as I mentioned earlier in the previous slide. It does show an action potential, but here it spontaneously depolarizes. It didn't need an external stimulation. The tissues spontaneously depolarized, hit the threshold, and generate an action potential, which is a rapid shift upwards to the more positive. And then it repolarizes down, and then again spontaneously you get a slower type of depolarizing until it hits the threshold, and again an action potential. The reason for this is, now what you have is a negative value, which is a negative membrane potential, which is generated by the fact that you have the sodium-potassium pump constantly pumping 3 sodiums out and 2 potassiums in. And this is true for all tissues, but in the heart you have these 3 sodiums out, 2 potassiums in, so the inside becomes more negative. And here it is, down at minus 60 here. However, the permeability to potassium starts to drop. That's the spontaneous characteristic of this, that's the spontaneous depolarization is because the permeability of the membrane to potassium starts to go down. Now you should realize that if the sodium-potassium pump is constantly pumping 3 sodiums out and 2 potassiums in, then you have a higher sodium concentration outside of the cell than inside, and a higher potassium concentration inside than out. Now, if the permeability to potassium is dropping, then the leaking of potassium going outwards because of the higher concentration on the inside, that's constantly sinking. Then potassium tends to be staying inside. Meanwhile, it has a low permeability to potassium, but there is some permeability to sodium, and sodium concentrations are quite high outside of the cell because of this sodium-potassium pump pumping 3 sodiums out. So there is a slow leakage of sodium back into the cell, causing, that's the real cause here of this depolarization. A low potassium permeability, but a slow leakage of sodium back in. So potassium's not leaking out, but sodium is leaking in. Therefore, it's the charge, the membrane potential is depolarizing, and it hits threshold. And when it hits this value of the threshold value, then calcium channels suddenly open, and you get these calcium channels opening here, causing a big increase in the permeability to calcium, because when channels open now it's permeable. Calcium is higher outside than inside, so calcium rushes in, and you get this rapid movement up towards the more positive, up this action potential. Now, once you get this sudden opening up of calcium channels, it is followed by a closing of those channels, so calcium permeability suddenly drops. Okay, so calcium stops rushing in, and potassium channels open. Potassium permeability rises. Remember that potassium is higher inside the cell than outside because of the action of the sodium-potassium pump. So now potassium starts to leak outwards. Okay, so what you have is that calcium permeability is no longer high. The calcium channels are closed. Calcium is no longer getting into the cell. Potassium is now leaking out of the cell. The cell inside is going to be repolarizing under those conditions. And then you get a reversion back to, again, that slow leakage of sodium in with a low potassium permeability, causing this slow depolarization to threshold. And then again, calcium channels open, and calcium is higher outside of the cell than inside, so calcium rushes in. Then the calcium channels close, so calcium stops rushing in. Things are getting more negative. The potassium permeability rises, meaning that there's more potassium leaking out. Things are getting more negative because there's less positive charges inside. In this way, you get this these impulses being generated by that autorhythmic pacemaker tissue. You get these action potentials being generated. Now that autorhythmic pacemaker tissue is found in this location, right up here in the wall of the right atrium. It is called and is initiated, the tissue is called the sinoatrial node. It is the pacemaker of the heart. It's usually called the SA node, and that's how I'll usually refer to it, the SA node. So the SA node is constantly depolarizing, going to the more positive. It is generating action potentials, and the action potentials will spread their impulses. And they spread across the tissue of the atrium, all across the tissue of the atrium, throughout the atrium, triggering, okay, those action potentials are quite positive. They trigger these other cardiac cells to go to their threshold, right? They normally at their resting membrane potential, but now they'll go to the threshold and they'll get that impulse, and the response is they contract. And they contract as one, and the depolarizing spread of the depolarizing wave, the action potential across all of the atria on both sides is of course very rapid because of the intercalated discs with all those gap junctions that allows very free flow of ions. Not only does you get a rapid depolarizing wave across the atria causing them to be triggered to contract, but that depolarizing action potential also spreads down to another tissue here called the AV node, or atrioventricular node. And there is a slight pause, a slight delay is caused here at the AV node. It doesn't generate its own action potential and stimulate. It doesn't trigger action potential immediately. There is a delay, and that delay is quite important because you want a delay between the signal getting to the atria and the signal getting to the two ventricles, because the atria need to be stimulated and contract just before the ventricles get stimulated to contract. So there's a slight pause at the AV node. Depolarization initiates at the SA node, spreads across the atria, they contract, and spreads down to the AV node, and there's a slight delay, and then it gets distributed down and through into the ventricles and throughout the ventricles along this pathway. It goes down this track called the bundle of His, and I'm going to do it in a different color, down through the bundle of His and the bundle branches that you see here, and spreads further throughout the whole ventricle. All of the area of the ventricle spreads all through along what are called Purkinje fibers. So the depolarizing wave that can stimulate the muscle, the normal cardiac muscle cells to contract, and they contract as one because they're all connected through as a functional syncytium, a single unit. That depolarizing wave initiates first at the SA node, spreads across the atria, down to the AV node, there's a slight delay, and then down the bundle of His, down along the bundle branches, and then the Purkinje fibers, spreads all throughout the ventricle, and the ventricles contract as one. And that's what causes this autorhythmic pacemaker activity of the heart. You can cut all external nerve innervation into the heart, and the heart will still beat. And that is why you can successfully transplant a heart, because of this kind of autorhythmic pacemaker tissue ability that resides in the sinoatrial node pacemaker. Now that initiation of a action potential and distribution throughout the heart is initiated, as I said, at the SA node, and it generates typically impulses. It depolarizes to threshold about 75 times per minute and generates a normal sinus, what's called a normal sinus rhythm of the heart, about 75 times per minute. The AV node, atrioventricular node, remember, delays the impulse by a brief period so that it gets delayed before it gets to the ventricles, so that the ventricles contract just a little bit after the atria. And that delay is 100 milliseconds or 0.1 seconds, same thing. So there's propagation of that depolarizing wave from the SA node to the AV node and then down to the ventricles along the bundle of His and the bundle branches and Purkinje fibers. And that's that propagation or carrying of the impulse along, as I said, bundle branches, and the bundle of His, the bundle branch, Purkinje fibers, so that it spreads all through the ventricle, and the ventricles contract all as one. So the cardiac cycle is that first the atria here contract, ejecting whatever blood they still contain down into the ventricles, and then because of the delay at the AV node, the ventricles contract slightly after. And that is what's shown here in this diagram. I just want to tell you that when you look at the ECG, please understand that what's shown in an ECG or electrocardiogram is what can be measured at the surface of the body. It is a measurement and a graph generated based on voltage changes of all of the tissue in the heart. Okay? So, and the more tissue you have depolarizing, the more effect it will have. And also the direction of the wave of depolarization can also affect that, what can be measured at the surface of the body. Okay, so the ECG is measurement in changes in voltage that detected from the heart, but measured at the surface of the body. And what I'm going to be showing you is that when you look at a normal sinus rhythm, and a normal sinus rhythm is shown here, a single cardiac cycle is called a PQRST, followed by another PQRST, that again and again and again. That's called the normal sinus rhythm, all of that. What you see here represents what's going on in all of the tissues of the heart sequentially. And I'll show you how that reflects, which place depolarizing in the heart is shown at the surface when measured with the ECG. Okay, so what you see here initially is the initiation of an impulse at the SA node, right there. And that causes what that initial wave is, which is called the P wave. So it's that first initial depolarization spreads, of course, across the atria and down to the AV node. And that contraction, that spread of that depolarizing wave, which triggers depolarization to threshold of all of the cardiac muscle tissue, which was at its resting, typical resting membrane potential until it gets triggered, and then you get an action potential, and it goes back like that. So, that's what I just drew that's what's happening in the cardiac muscle cells, the normal ones, not the autorhythmic SA node. So they're being triggered to contract, and what that can be measured, detected at the surface of the body using the ECG, and it's detected as a P wave. That spread of depolarization across all of the atrial tissues detected as a P wave. Then it spreads with a slight delay at the AV node, then it spreads down the bundle of His and the bundle branches and along the Purkinje fibers, and you start getting a spread through the entire ventricular tissue, and all of the ventricles, cardiac muscle tissue gets excited, and you get triggered contraction, and that's shown in this QRS complex here. And then you get repolarization, which is of the ventricles, which is shown with this wave, which is called a T wave. Okay. So depolarization of the ventricles is the QRS, and then repolarization of the ventricles is the T. Now, as I said, you do have external nerves. You do have an innervation of the heart from outside, extrinsic, outside, external, innervation, connection with a nerve input into the heart. Remember, I told you that you have involuntary excitable tissue, which is cardiac muscle. I'm just going to put C, cardiac muscle tissue, involuntary, and smooth muscle tissue, and glandular tissue. They all are controlled by the autonomic nervous system, and there are two divisions to the autonomic nervous system that affect these kinds of tissue. And glands. All of these are involuntary effector tissue that gets excited or controlled by the autonomic nervous system, and that there are two branches: the sympathetic division of the autonomic nervous system and the parasympathetic. And the sympathetic acts when you're excited. This is the fight or flight response, and the parasympathetic is when you're relaxed. So sympathetic stimulation of the heart is actually, it triggers a increase in the heart rate, because let's face it, when you're excited, stimulated, fight or flight response is triggered, sympathetic division of the autonomic nervous system is triggered, your heart's going to speed up. You need to be able to pump more blood at that point because you might have to run, you have to. So this accelerates the stimulation of the heart. And that is coming out of the brain stem, sort of the brain, it's coming out of the medulla oblongata of the brain stem. The brain stem really controls the most fundamental basic responses in the body, breathing, heart rate, things like that. And it stimulates, so you have a pathway. It's a complicated one. I wouldn't worry too much about it, but it's, don't worry about all this stuff other than the fact that it stimulates the SA node and stimulates the AV node and stimulates the ventricular tissue shown there. That's innervation, sympathetic fibers causing stimulation, sympathetic innervation signals coming in. That's when you have a sympathetic increase in sympathy. If you're calm and you have more parasympathetic stimulation, okay, parasympathetic, that's going along here, nerve that comes down and has a inhibitory effect, not a stimulatory, but an inhibitory effect on the AV and on the SA and AV nodes. Okay, as I was saying, in terms of what can be measured using a electrodes at the surface of the body is the electrode. You can measure using an electrocardiograph. You can measure the depolarization of the entire heart tissue that's undergoing these changes in charge, internal charge, depolarizations and repolarizations. Depolarizations going to the more positive, repolarization going back down to the more negative. And it's determined not just by positive and negative, but also by amount of tissue involved and by the direction. Don't forget that the wave of depolarization across the atria is very rapid, but nevertheless in one direction. And the wave of depolarization, of the ventricles, even though it's very fast, is in a different direction. So things can get very complicated. But in the end, what you are seeing when you measure at the surface is you will get a PQRS-T shaped wave. The P wave represents atrial depolarization, going towards the more positive. The QRS complex is representing ventricular depolarization, and the ventricular repolarization is represented by the T wave. So we have atrial depolarization P, ventricular depolarization QRS, and ventricular repolarization T. Remember, tissue, like in the atria, depolarizes, goes to more positive, then repolarizes, and you don't see that here in this graph because it's hidden under here, under the big QRS, right? The ventricles have much more tissue, so you can't actually see it. It's occurring in this time period. Atrial repolarization is occurring at the same time as ventricular depolarization. And the ventricles have more tissue, so it's covered by, hidden underneath this big QRS complex. And then ventricular repolarization is shown by this T wave. I'm going to stop here. This is the end of Unit 6, Part 2.